Thanks everybody for joining us. So I'll go ahead and introduce Dr. Rodrigo Mendes. Rodrigo is a research scientist at Embrapa and a professor in the postgraduate program at the University of Sao Paulo in Brazil. His research focuses on decoding the cry for help hypothesis to uncover how the rhizosphere microbiome comes to the rescue of plants facing nutrition scarcity, drought, and also to pathogens. Rodrigo holds a degree in agronomic engineering and a PhD in genetics and plant breeding from the University of Sao Paulo. And from 2015 to 2022, he served as the head of research and development at Embrapa Environment and has held positions in many familiar institutes around the world, including Wageningen, the University of California, Berkeley, the University of Lausanne, Rothamsted, and the University of Cambridge as well. So we're deeply grateful, Rodrigo, that you've joined us today. Thank you very much, and I'll hand over to you for your seminar. Thank you.
Thank you, James. Thank you for the invitation. I'm really happy to have this opportunity to share my view on the rhizosphere microbiome as the second plant genome. So maybe you recognize this small device in your screen. It's a 5 centimeters long device, which is a very good version of one of the first microscopes. It was designed by the Dutch Anthony Leeuwenhoek in 1670s. So this is the 17th century, long time ago. You have the screw, a small needle, a small hole on you put the lens, and then he used a candle as the source of light to observe different samples. And then, of course, you can imagine when Anthony Leeuwenhoek for the first time collected a drop from a lake, from a pond, and then put in the microscope to observe. So basically, a new universe was opened in front of his eyes. He was able to see microbes for the first time. Of course, at that time, he didn't call that microbes. He used the term animalculus, which means small animals. And then he drew the structure of different and diverse small animals, the microbes. It's interesting to think that the microbiology was born, look at the microbes interacting in a complex mixture of species and living beings. Later on, the microbiology changed a little bit the approach, the way we use it to study microbes, because we were using the approach of isolating microbes in petri dishes, and then we look at the microbes in an isolated form. But more recently, so basically 350 years later, the microscope was reinvented. And this is one of example of a sequencer, DNA sequencer. So basically, as in the 17th century, with this technology, we can go back again in environmental samples and then look at the microbes working and living and interacting in the context of the environment. So what did change when we started using sequencing to study microbes? There are many impacts on the microbiology. And one of the most important one is the impact on the way we organize life in the world. So basically, using information of sequencing, we changed the topology of the tree of life. And then we also added new microbes, new lineages. it's interesting to note that in this figure here, all these lineages with these red dots, they lack in isolated representatives. So basically, we don't store those microbes in the freezers, but we store the information of those microbes in computers. So what if we use this powerful tool, this reinvented microscope, to study the interactions between microbes and plants in the rhizosphere? So what I'm going to show you, it's a study where we investigated how the microbial community interacts with plants in the root system using metagenomics. To do this study, we used what we called disease-suppressing soils. I'm going to just explain you the concept of disease-suppressing soils, which is really interesting, and it's old knowledge. So more than 100 years ago, farmers and growers, they realized that in some specific fields, after an outbreak of a given disease, normally a soil-borne pathogen causing an outbreak of a given disease, if they insist in cultivating the same field using the same crop, a susceptible crop, in the presence of a soil-borne pathogen, there was a decline of disease over the years. They didn't understand what was going on, but now using amplicon sequencing and shotgun sequencing, we investigated the same phenomenon. So if you look at those two plots in your screen, on the left side, we have a disease-suppressive soil. So what you see here are healthy seedlings of sugar beets, and we inoculated a pathogen in those pots, in this case, Rhizosphere solani, which is a very common soil-borne pathogen able to infect many different crops in the world. But despite we have a susceptible host and a virulent pathogen, we don't see the disease. And the question is why? And the counterpart of a disease-aggressive soil is the conducive soil. It's what you see in the right-hand side of your screen. In this soil, we sowed the seeds of sugar beet, and then we introduced it to Rhizosphere solani, the pathogen, and the pathogen was able to grow, to infect the root system of the seedling. They cause a black lesion, and this phenotype, as we call it, dumping off. They kill the seedlings. And what makes this even more interesting is that the soil in these two pots, they're the same. They share the same physical and chemical properties. But in one case, the plants are naturally defended against the pathogen. So why? And then, of course, let's look at the microbiome associated to those plants. So this is the bioassay that we use it to try to understand the mechanisms behind the natural disease suppression. So again, if you take a suppressing soil and then you introduce the pathogen is not able to cause disease, or it's able to cause just a little amount of disease. So this is the first bar in this graph. A limited number of plants are infected by the pathogen. But if you do the same experiment with a conducive soil, more than 60% of the seedlings are affected and are killed by the fungus. So then we have these two contrasting phenotypes, and there are two ways that we could use as a proof of concept to demonstrate the microbiological nature of protection. So the first one, if you take the suppressing soil and then we heat treat the soil for 80 degrees Celsius for 30 minutes. And then we repeat the bioassay. This is what we have got. This is the suppressive soil, heat treated, and then we repeat the experiment. So the disease incidence of this suppressive soil is comparable with the conducive soil. So basically, using temperature, we can transform a suppressive soil in a conducive soil. So what is the rationale behind that? So when we are using temperature, or we can use irradiation, light, like gamma irradiation, and then we observe the same change. We are impacting the microbes in the soil, and then the soil and the plant growing the soil is not able to recruit beneficial microbes to protect against the infection. So this is the idea. There is a second strategy to demonstrate the microbiological nature behind this phenomenon, which is the soil transplanting. If I take a portion of suppressive soil and then I transfer in a conducive soil, I'm able to reduce the disease incidence. So when we compare this C bar, which is a conducive soil, with this CS bar, which is conducive, amended with suppressive soil, 1 part of suppressive soil to 90 parts of conducive soil, basically 10% in a mix, I'm able to significantly reduce the disease incidence. So what is the rationale behind that again? When I transfer the soil, I'm transferring beneficial microbes that are able to somehow restore, to some extent, the natural protection obtained by the plant. So this somehow impacts the way we see the plant, because we have to take into account the microbes associated with the plant to understand how the plant performs in the environment. So basically, we can take 3 assumptions considering the plant in association with the microbiome. First, we can see it as a superorganism. It's not only us or an insect or a plant, it's us plus the microbiome associated to us. So this view, looking at the microbiome as an extension of the genome of the host. We can also call this a holobiont, which is by definition a unit of evolution. This is the first assumption. Look at the plant and microbes as a superorganism. Second, the host is able to modulate the microbiome. The microbes associated with the plant roots, at least a fraction of those microbes are selected and actively recruited by the plant. There is a control of the host to select microbes living in association with the host. And then the third assumption, I would say, the host uses outsourced services provided by the microbiome. The host can rely in specific functions, in this case, plant defense, that are available as ecosystem services in the microbiome. So in nature, we can have many examples on this type of interactions. For example, ruminants, they rely on the rumen microbiome to break down the fiber present in the diet and then get energy from the fiber. There is this dependence for specific functions. And when we think about the rhizosphere, which is the soil surrounding the root systems of the plants, we can see it as the gut inside out. So that's why I brought a small mice in the screen to tell you that there are striking similarities when we compare microbiome functions across different animals or plants or insects. If we think about the gut microbiome or the rhizosphere microbiome, so in both, this living layer of microbes in the interface between the host and the environment plays a key role for many specific and important functions related to health, development, and growth of all organisms. So with this view of trying to understand the plant as a superorganism, we use it, and this is just the summary, a model summarizing the findings of different studies, where you used mostly amplicon sequencing, shotgun sequencing, metatranscriptome sequencing to try to understand what is going on below ground. So the story here is that when a seed of sugar beet sowed in the soil germinates, the seed attracts some specific beneficial microbes as releasing chemical compounds attracting those microbes. It's a chemical language between the plant and the microbes. But it's interesting to note that the same language is understood by the pathogen, which is also in the soil in this case. So the pathogen, which could be sleeping around, the pathogen realizes the presence of a plant growing in the surrounding based on the chemical released by the plant, and then germinates and grows throughout the soil to meet and infect the root system of the plant. But in disease, in conducive soil, what we would expect? This is in the phytopathology we call the disease triangle. We have a susceptible host, a virulent pathogen, and environmental conditions where they can interact and result in disease. This is what we would expect. Pathogen, susceptible host, disease. But we don't observe that in disease suppressing soil. So when the pathogen grows and approaches the root system, there is a reaction by the plant responding to the presence of the pathogen. And there are two important changes in the microbiome. First, there is enrichment and activation of specific bacterial families. They increase in abundance in the presence of the pathogen. And secondly, the presence of the pathogen activates functions related to bacterial response to the stress. And the outcome of those changes in the rhizosphere is that the plant's roots are protected from the fungal infection. Right? It's a natural process. And then we can see this in the context of the cry for help hypothesis. So under a biotic stress, when the plant is threatened by a pathogen, the plant modulates its physiology, releasing different chemicals in the soil, communicates with specific members of the microbiome, and those members of the microbiome are able to rescue the plant against the pathogen. So we have learned, and I'm going to highlight 4 points in this model that can help us or give us some possibilities on how to manage or how to explore this natural phenomenon in the rhizosphere microbiome. So the first lesson, microbial diversity is key. When we compare suppressive soil with conducive soil, one of the big difference is that suppressive soils, they are able to harbor a higher microbial diversity. And this makes sense in the context of the cry for help hypothesis. So when a pathogen gets close to the root system and the plant cries, we need microbes to respond to its cry. And if you are in a soil with low microbial diversity, you don't have the key players to protect the plant. And this is what we get in a conducive soil. So first, microbial diversity is key for plant protection. Second, beneficials. If I can identify specific members of the microbiome that increases in abundance in the presence of the pathogen, I can target those beneficial microbes, isolate them, and as a proof of concept of experiment, I can use those beneficial microbes to inoculate in a conducive soil, and then those microbes will protect the plant against infection. So, and then we run a series of experiments in doing that, selecting those members of beneficial microbiome that increased in the abundance in the presence of the pathogen, inoculating them in a conducive soil, and they were able to defend the plants. The third highlight, the communication, it's all about signals and chemicals. Maybe we don't need to add new microbes in a conducive soil, but we can play with signals. And again, as a proof of concept, we learned in this model that the oxalic acid produced by the pathogen is able to trigger the reaction in the plant to change and shift the composition or the relative abundance of key members in the rhizosphere microbiome. So what we did to demonstrate that, we took a conducive soil and then we added not the pathogen, but the oxalic acid to trigger this type of a reaction. And then we added the pathogen. And we observed that the addition of oxalic acid also gave, to some extent, protection against the pathogen. This is also a way to go in terms of how to manipulate the microbiome in crop systems. Maybe we can use signals that can trigger beneficial responses from the microbiome. And the fourth one, efficient plants. So this is important because when we look at all these interactions going on below ground, who is orchestrating all this is the plant. So the question is, so can we have more efficient plants in terms of communication with the soil microbiome? Why? Because we know that the modern plant cultivars we use today, they're quite different when we compare them with their wild relatives. And when we did plant domestication and plant breeding, maybe we have impacted or we have left behind important plant traits that are important for the plants to communicate with the microbes. And then we are able to test that in an initiative called Back to Roots. So nowadays there are many groups working on that, comparing modern plant cultivars with their wild relatives, testing this hypothesis. When we selected plants from the natural center of diversity, and when we used plant breeding and the selection pressure we put in some specific traits like productivity, stability, maturation. So have we selected against the traits that were important for the plants to communicate with the microbes? And then I'm gonna finish showing one example in wheat, where we compared modern wheat cultivars with land races collected from where we have the center of origin of wild wheat in Iran, Turkey, and Pakistan. We use it, just showing here the data on the amplicon sequencing to access the bacterial community, the fungal community, and the cercozoa community. This is an ordination analysis. We tested landraces of wheat and modern cultivars in two types of soils: in agricultural soil with history of wheat cultivation, and in a more virgin soil collected from the forest, but the results are the same in different soil backgrounds and very consistent. If you look at the bacteria, for example, we see a cluster of samples with different shades of blue. Those are all samples coming from modern wheat cultivars. In another cluster of samples, we see all these landraces of wheat. So if we grow landraces and modern cultivars under the same conditions in the same soil, they assemble a very distinct composition and structure in the rhizosphere microbiome. And this is not only true for the bacterial community, but it is also true for the fungal community and for cercozoa community. So they are able to communicate with different microbes. And to give you one step further, we did this co-occurrence analysis or network analysis to show the potential interactions when we compare the fungal community, bacteria, and cercozoa. When you look at a figure like that, it's sometimes it's too much, yeah. But actually, there are many mathematical parameters behind this figure, like the number of nodes, if the connections are positive or negatives, the density, modularity, centrality. But I'm going to show you the results on the modern wheat cultivars. These are landraces, old material. And now you see the same analysis for modern cultivars. So we missed something, right? It's much more simplified in the modern cultivars. Let me go back here again. Landrace of wheat and improved lines of wheat. So apparently, the modern plants, the ability of the plants to communicate with the microbes was mined with domestication and plant breeding. So can we, let's say, rewild modern cultivars to explore those natural benefits in the interaction between plants and the soil microbiome? 3 years ago, two groups, one working in tomato in the Netherlands and another one in barley in Scotland. They showed, I think it was for the first time, the link between a genetic trait of the plant and the recruitment of specific members of the rhizosphere microbiome. So this means that there is a genetic marker that is correlated with the recruitment of specific microbes. So this opens the way to explore or to do plant microbiome breeding in a way that we can select traits recovered from ancestors or wild relative and reintroduce in modern cultivars in a way to explore the ecosystem services provided by the rhizosphere microbiome. And to finish, this is my last slide, just to give you so what it's coming in the next 10 years in terms of exploring the rhizosphere microbiome and plants using sequencing technologies. So I selected here 1, 2, 3, 4, 5 points. So first, I think we're going to explore new branches of the tree of life. In agriculture, we are still limited to work always with the same species, same microbes. I think we're going to learn how to introduce new species of microbes and bring into farm settings. Secondly, we're going to learn more from what to how they are doing that, and they're understanding the underlying mechanisms of specific functions like nutrition, tolerance against abiotic stress, that are as they go through the microbiome. Plant microbiome breeding, I think we're gonna be able to obtain modern cultivars more efficient in explore these beneficial traits present in the microbes associated with the plants. I also suggest that we're going to use a more trait-based approach instead of a taxonomical approach. More important than who is there, it's what they're doing. We're going to explore how to assign specific functions. We are going to be less concerned about the names of the microbes, but with the function. This is really important in microbial ecology, as we have what we call a functional redundancy. So we can have completely different structures of the microbiome, but they can work in similar ways. So trait-based approach. And of course, we're gonna also get help from artificial intelligence in terms of using complex microbiome information to make predictions in agriculture, and also to help us how to obtain better microbiome assemblages. So I will stop here, and I'm happy to discuss and to take some questions. Thank you very much again.